Okay. Okay, folks, pep on up. We got the glycogen metabolism going. Nobody even caught the bad pun? Shit. All right, so chapter 21, glycogen metabolism. Obviously, this person was resting to recover. This is what the glycogen granules actually look like in the liver cells, by the way, which is quite interesting. Why? He has the little mat under it. Yeah, no, that's just weird. Yeah, it's very staged. I have no idea. Yeah, is the cat towel or a mat? Yeah, I think it's a yoga mat. Yeah. Though, to be fair, the positions I've seen students take and they're catching a few Z's between classes don't look any more comfortable, do they? Yes, indeed. Maybe he needed extra safety and he felt the dumbbell would keep him safe. I don't know. Maybe. Yeah, he has a worry that he's going to float off. Anyways, glycogen metabolism, regulated release and storage of glucose in multiple tissues. So the glucose cannot be stored because high concentration of glucose will actually disrupt the osmotic balance of the cell. This would actually cause cell death or damage. Glycogen is a significantly less osmotic, active, and it's going to be highly branched polymer. This can be rapidly broken down to yield the glucose, which can then be removed so you don't build up osmotic pressure. It's present in bacteria, archaea, proteus, animals. Its controlled release maintains the blood glucose concentration between the meals. And so typically what would happen is you're going to peak at your blood glucose after your meal. It's going to lower throughout, and then you'll start releasing it from the glycogen after that to keep you at a level playing field. To be honest, it doesn't actually work all that great in most people. So, yeah, for various reasons. But it's a good source of energy for sudden strenuous activity, as it can be metabolized in the absence of O2. So liver and muscles tissue are going to be the primary storage sites for the glycogen. The glycogen is going to be present in your cytoplasm. It'll appear as these little granules of multiple glycogen molecules. And so that's what it looks like zoomed out. That's what it looks like zoomed more in. And you can see the little granules that clump together, right? And those are the glycogen. Ooh, so beautiful. Okay. So this is essentially what an individual glycogen molecule looks like, in abstract a bit. It has 12 layers of glucose molecules. It can be as large as 40 nanometers. It will contain 55,000 glucose residues, which would be the monomers. It has a single glycogen and protein. That single protein is at the core right there. Can you see that yellow thing? Yeah. Okay. So most of the glucose is going to reside in glycogen. It's going to be linked by the alpha-1,4 glycosidic bonds. You may remember your alpha-1,4 glycosidic linkages right here. Those are the straight chains. Okay. The branches are going to be these joinders right here. They're going to be about every 12 residues. They're going to be created by the alpha-1,6 glycosidic bonds. And that's the alpha-1,6 right there. And that's the branching. Okay. We've actually kind of talked about this a little bit before, but we're just going into more depth now. These right here are the non-reducing ends, by the way. Everybody good so far? Fantastic. Notice, by the way, this has an R This could be more of this molecule, or it could be linked back. So glycogen degradation and synthesis. The glycogen degradation is going to consist of 3 steps that will ultimately produce glucose 6-phosphate. Remember, glucose 6-phosphate can be metabolized by glycolysis. It can be converted into free glucose in the liver for release into the blood system, where it can be transported all around, say, your brain or your heart, right? Or your muscles, maybe. This could be processed by the pentose phosphate pathway. This would give you NADPH and ribose derivatives. All the information we got from the pentose phosphate pathway chapter, that all applies here, okay? Glycogen synthesis is going to require several steps. It's going to take place when glucose is abundant and glycogen is depleted. Anybody know what people typically call the glycogen storage? If you have a bunch of glycogen storage and it's visible from the outside of your body. It's your beer gut. It's that part right over your liver right here that's a little harder. Yeah. And the reason that's hard is actually because it's glycogen buildup. Yeah. That's why it's so solid and hard. It also is in your muscles too. Though most people are active enough that that's not a huge thing. But yeah, it does build up there as well. So the glucose 6-phosphate, this can have many metabolic fates, of course. It's a pretty ubiquitous molecule throughout many of these pathways we've been talking about. Right here is your glycogen. So we're going to chop one of these glucose 6-phosphates off of that glycogen. So your glycogen is going to go to N-1. Everybody's comfortable with that terminology? Fantastic. That's that polymer terminology we haven't seen for like a chapter or 2 now. Okay. The glucose 1-phosphate then comes off there. By the way, the enzyme is glycogen phosphorylase. By the way, phosphorylase. What do you think that means? It phosphorylates, right? Over here, we have then glucose 1-phosphate. This is going to have the enzyme phosphoglucomutase. That's going to give you the glucose 6-phosphate. The glucose 6-phosphate is a much more ubiquitously useful molecule. And so we have the mutase to rearrange that. This is then going to go into a variety of different pathways. It could go to pentose phosphate pathway through that same mechanisms we looked at before. And that could give you things like ribose and NADPH, right? It could go to glycolysis, where it would go to pyruvate eventually. If you have oxygen available, that would go to CO2 and water, which would be your citric acid cycle, right? Electron transport chain. This also could go to lactate, which is your lactic acid buildup because you don't have aerobic conditions. This right here could also just give you glucose. So we could transfer it, process it more to glucose in the liver, typically. The enzyme that does that is glucose 6-phosphatase. Again, we're going to be taking that phosphate off, so glucose 6-phosphatase. That's going to give you glucose, release that into the blood, and that can go anywhere in the body and do all the things that glucose can do, right? You do need a minimum level of glucose in your blood, and this is typically how you maintain that level. Okay? Anybody know people that have an error that they cannot maintain glucose levels? They have to have a little Snickers that they eat all the time. So they always have to get glucose in their blood. Huh? Yeah. It's without the glycogen storage and then releasing that, it's actually incredibly dangerous to go without. Because remember, your brain and your red blood cells, they only use glucose. So you run out of that, you're going to die. Glycogen phosphorylase. So this is going to be phosphorylase. This is going to cleave the glycogen via the addition of an orthophosphate. Phosphorylasis, not to be confused with phosphorylase. Everybody hear that? Phosphorylasis. Lace. Okay. You can actually split this up if you have difficulty. Phosphorylase. Phosphorylysis. Okay? Okay. By the way, do actually try to say these words at some point before you try to go to like an interview or a med school. Okay? Because it's not always fair, but people do judge you on your ability to talk the lingo and say the words, right? And I'm going to tell you, you have a bit of a, not a huge one, but a small disadvantage in that, okay? Which is that you have a slight Southern accent, most of you. And that slight Southern accent is not the accent that these were done for, right? It doesn't sound right, okay? So you do have to make sure you actually say it. It worked, okay? The glycogen plus the orthophosphate, this is right here. Again, this would be in residues. So we cut one of those glucose off with this phosphorylase, and this is going to give us our glucose 1-phosphate and glycogen with one less residue, right? So the phosphorylase, is everybody okay if I say phosphorylase? You can understand that fine? Nobody has any issues? Okay. Mechanism. The phosphorylase catalyzes the sequential removal of your glucosyl residues from the non-reducing ends. Remember those non-reducing ends over on the left side of the figure, right there. Everybody see it okay? Okay. They're red ones. Okay. So the orthophosphate will split the glycosidic linkage between the C1 of the terminal residue and the C4 of the adjacent one. The alpha configuration of the C1 is actually retained. Everybody remembers alpha, beta on the sugars? These linkages. Beta would have been up, right? Alpha would have been down. Okay. So the glucose 1-phosphate, this can be converted to glucose 6-phosphate by the phosphoglucomutase. And that's this right here. We got the phosphate, orthophosphate coming on. Notice again, these are the alpha orientations here for those. And it maintained that. And that's the glucose 1-phosphate coming off, which will then be rearranged. The phosphorylase is going to be a dimer of identical subunits. Each of the compactly folded into an amino terminal domain containing a glucogen binding site and a carboxyl terminal domain. The active site is going to exclude water. This will save the ATP that's required to phosphorylate the free glucose. Excuse me. So both the glycogen substrate and the glucose 1-phosphate product have an alpha configuration at your C1. This actually suggests that carbocation intermediate is formed in these cases. The glycogen phosphorylase requires the pyridoxal phosphate, this is PLP typically, as a cofactor. The aldehyde group, PLP, is going to form a shift-base linkage with the lyse residue of the phosphorylase. Yes, same as we just had talked about last chapter, the shift-base double bond nitrogen. I know, you were wondering where the damn organic chemistry was involved. There you go. That's so organic chemistry, right? Made you go right back to last semester. The glycogen binding site is going to be 30 angstroms away from the catalytic site. It's going to be connected by a narrow crevice that's able to accommodate 4 to 5 glucose units. It's going to enable the enzyme to phosphorylize many residues without having to dissociate and reassociate. Remember, one of the most time-intensive things for many of these enzymes is not actually the reaction, right? What do we usually consider a perfect enzyme? What do we measure that in time? The time it takes to get attached, right? to have substrate come to it and get attached. Okay? So this enzyme is actually really impressive because you can have a number of glucose in this active site, and then you do have very, very little time in actually having those reactions happen, right? It really cuts down on the amount of time. Imagine if it had to attach and reattach every time, right? You guys ever seen like a car assembly line versus a Coca-Cola bottling factory? That would be the difference, right? The car assembly line has to have people come on and off all the time, right? Whereas Coca-Cola bottling factory does like thousands of products per second. Because they don't have to have anything coming on and off. Okay, so anyways, really neat active site here. This is the glycogen phosphorylase, the homodimer right here. It has the PLP group in each of the active sites here. These are catalytic sites right there, there, and there. Wow, the gray one is so much easier to look at, isn't it? Right there. And then here it is on the yellow one. Can everybody see that okay? The little black thing in there. Okay. Yeah, this is the glycogen binding site, by the way, right there. And there's the glycogen binding site on this side. Here's your N-terminal domain, C-terminal, right there. This is the binding site zoomed in right here. And we can actually see the orthophosphates right here. Right here's the lyse, right there, and there. Okay. This is actually that shift-based linkage. And so here's the PLP. It's making that shift-based linkage. Again, there's the lysine making the shift-based linkage. And that was right over here. Can everybody see that? Phosphate right there, PLP, lyse, shift-based linkage. Okay, so this is the proposed reaction right here. And here we have our sugar, glycogen currently, will be a sugar. This is going to then have the PLP right here, and this is that orthophosphate that came on. What's going to happen then is the orthophosphate is going to react with this oxygen with the rest of the chain behind it. This is then going to put a hydrogen on here. That will free that as the alcohol. This then is going to leave behind a carbocation. This is that super reactive intermediate right there. And what's going to happen then, this leaves the site. This super reactive carbocation is going to attack one of these oxygens, and that's going to attach that phosphate on there. That's going to cause that exchange of the hydrogen back to the PLP. And notice that PLP is actually acting as that shift base that you oftentimes see in these carbocation type reactions, or carbanion reactions in the other case. Okay? Because that shift base has a very good ability to take on multiple states where that charge can be shared with that nitrogen, right? And that's what you're seeing getting taken advantage of here, which is pretty cool. All right, so that glycogen phosphorylase, this can only cleave the alpha-1,4 glycosidic bonds. Hey, wait a second. Those are only straight chains, aren't they? So what happens when it gets down to a branch? Okay, so let's just quit here and you won't know this ever. Now I got you curious. Okay, so the transferase, this is going to be what shifts a small block of 3 glycosyl residues from one outer branch to another. This is going to expose a single glucose residue that's joined by an alpha-1,6 glycosidic bond. The alpha-1,6 glycosidic bond, which is the branch, right, has the alpha-1,6 glucosidase. This is going to be what hydrolyzes the alpha-1,6 glycosidic bond. Notice the wording there, it hydrolyzes it, right? Which means water attacking and splitting that. The debranching enzyme is a bifunctional enzyme in eukaryotes that's going to contain transferase and the alpha-1,6 glucosidase activities. So notice that we had the transferase we just talked about here, shifting the rest of that chain, right? Okay. And we have the alpha-1,6 glucosidase. This together, these two activities, are joined together into a single debranching enzyme when we're talking about eukaryotes. Okay? By the way, why are we shifting these things off? Why don't we just cut down until we get to it? Ooh, I'm making you do critical thinking again. Damn it, Dr. Clausen. Why? Don't look at me. You tell me. I don't know if it requires less energy or not, but there's a very good reason that you can't possibly not do it. Okay? Does this just take one glucose residue into it at a time? No, it takes about 4, right? So if you have 3 left, and then the alpha-1,6 linkage, you can't actually get it in there, can you? So it physically can't reattach to that point. Okay? It would greatly struggle. And yeah, energetically is why it can't attach to that point, because it wouldn't have the 4-branch attachment. Yeah. But yeah. It's actually a size. It'd be like trying to cut, have you ever tried to cut that little tiny fringe off with a big pair of scissors? And you literally can't fit it in between the two? Yeah, it'd be like that, right? Okay, and so the hexokinase phosphorylates the glucose if the glucose will enter the glycolysis or pentose phosphate pathway. This right here is that graphically done. So this would be that in protein core for your glycogen right here, okay? And then we have our glycogen right here. This is the straight chain. This is the branch. And after a branch, you're just a straight chain again, right? Is everybody okay with that? Okay. And typically, we branch how many? Every how many? About 12. Okay. Okay. And so what's going to happen is we're going to chop, 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 chop. We're chopping down, right? Okay. And then we get to the last 4 up to a branch right here, right? And this last 4 is no longer going to be able to go into the phosphorylase reaction site. We're going to have to stop chopping with that. What's then going to happen is this is going to utilize this enzyme right here, which is your debranching enzyme. Okay? Now notice that's bifunctional. That does both of these. And so the transferase is going to transfer these 3 here to a branch, to another branch, which it will be a chain, straight chain. And then this, the alpha-1,6 glucosidase, is going to then hydrolyze this bond and release the glucose. Now notice it's not the phosphorylated glucose, is it? It's not the glucose 1-phosphate, glucose 6-phosphate either. Okay? Just straight up glucose. Right? And that just enters the normal pathways glucose would normally enter. It's just not as far along in the pathway, right? And so we'd have to set that up. The glucose 6-phosphate, on the other hand, is part of the way through the pathway. So, and it can enter many different pathways just right there. Is everybody good with that? Fantastic. What if your branch only had like 2 or 3 glucoses on it? Is there any problem there? No. No, you just added 3, right? You're fine. Yeah. So, as long as you're not down to the last one, you're okay. And the last one would have gotten reacted with this, right? So, good. Doing good. So the activation of your alpha-1,6 glucodase, this is that activation right here. Again, we're doing the hydrolysis reaction for this alpha-1,6 linkage. And that gives you the water across there, which is the OH and OH, right? No phosphorol. Okay. Everybody okay with that? Questions? So the phosphoglucomutase is going to convert your glucose 1-phosphate into the glucose 6-phosphate. A phosphoryl group is going to be transferred from a serine residue at the active site of the phosphoglucomutase to the C6 hydroxyl group of the glucose 1-phosphate. This will form the glucose 1,6-biphosphate first. To restore the phosphoglucomutase, the C1 phosphoryl group of the glucose 1,6-biphosphate is going to be transferred to the serine residue. This will form the glucose 6-phosphate. Remember that in these cases, it's not usually as simple as just cut off the same one and move it. That's not something that can actually be done easily, right? Because then you'd have to attack a fresh substrate, wouldn't you? You wouldn't have any energy remaining. You would have lost all that. So what typically happens is they take one of these, they phosphorylate it, and then you cut the phosphorol to recover energy, right? Okay. And so right here we actually see that serine, it's bonded to a phosphoryl group here. We have the active form of the enzyme. The glucose 1-phosphate comes in, and what we'll do is we'll phosphorylate this one right here. Okay? And so that will still then have the biphosphate at that point. So it will be the 1,6-biphosphate, 1,6. And then we were going to go ahead and have this serine, which is no longer phosphorylated, right? And so the 1-phosphate can then be transferred onto that serine. And that's the glucose 6-phosphate now. The glucose 6-phosphatase is going to hydrolytically cleave the phosphoester linkage of the glucose 6-phosphate in the liver. This is going to yield the glucose and orthophosphate. And again, this is just hydrolysis of that phosphate. Okay? And you do this to get the glucose. So you can put that in the blood then, allow it to transfer around to your various different parts of your body that might need it. The glucose 6-phosphatase is going to be absent from most other tissues. Your muscle tissue will actually retain that glucose 6-phosphate for ATP generation. The glucose is not a major fuel for the liver, and so it can afford to lose that fuel to the places that need it more, right? The free glucose is going to be released into the blood for use by the brain, red blood cells, and of course other tissues like muscle, etc. Okay? Oh, there you go. See, you need to tell your liver, ramp it up. Yep. Any tiredness today, you just need to go. Liver, ramp it up. Glycogen. Okay, so your glucose 6-phosphate, the glucose 6-phosphatase is going to be located on the luminal side of the smooth ER membrane. Your glucose 6-phosphate is going to be transported into your ER. Your glucose and orthophosphates are going to be shuttled back into the cytoplasm. The glycogen phosphorylase is going to be regulated by these the allosteric effectors that are going to signal the energy state of your cell. The reversible phosphorylation, this is going to be responsive to hormones. And this is actually the one that most doctors are more interested in, okay? Because this is the sort of stuff that's going to allow you to modulate different forms of diabetes and stuff like that. Okay, have responses that are more like you dieting, that sort of thing. Okay? Releasing glucose at appropriate times, even those out. This is also the part that usually gets fucked up, which is like really common in humans actually. You may notice that most of us suffer from low and high blood sugars at different points of the day, right? Yeah, super common screw up in humans. Very rare for somebody to not have that screwed up. Glycogen phosphorylase has two isozymes forms. We have the liver one and the skeletal muscle one. The dimeric phosphorylase exists in your two forms. Usually active phosphorylate A form, that's the active form right there. Again, this is the activated phosphorylated one. The usually inactive unphosphorylated form. This is your B form. Okay? This is one that's activated with phosphorylation. Both forms are actually going to exist in equilibrium between an active relaxed state and a less active tense state. So you'll have an equilibrium for this phosphorylase. A favors the R state. The equilibrium for phosphorylase B favors the T state. Okay. And so right here we actually can see this nicely in, you may remember that dimer that we looked at before, the homodimer. Okay. This right here is going to be the phosphorine residues right here. These are the active catalytic sites right there. This is the phosphorylase A. This is in the R state right now. This one right here is the phosphorylase B, and this one is actually in the T state right now. And it's not quite as obvious as some of the other enzymes we've looked at in this structure, but you can kind of see it if you pay really close attention to this parts right here. See those? Right there? Yeah. You can see it a bit easier. Unfortunately, it's not like hemoglobin and a lot of the other ones we looked at, which literally has like a hole that's it's open, it's gone. It's open, it's gone. Yeah. This one is not quite as easy because you can't see down into it, right? Yeah. But the two states, if you pay really close attention right here especially, become much more obvious. Okay, so the phosphorylase, again, this can be regulated via phosphorylation. And so what we see here is this phosphorylation right here happening. So this is the phosphorylase B forms right here. These are the active sites. So this is the T state. This one's the R state. Okay? Can everybody see those okay? Yeah. And you can kind of see it up here a little bit. See how this loop here is over here? And this loop comes up into it. Okay? And that's what that is. Right there, that came up into it, down, away. Okay. It's not super easy to see because, especially down here, I think this is incredibly difficult to see personally. The colors just do something to my eyes. Yeah. But, yeah. So anyways, that's the R and T state. Notice again, we have the phosphorylated, non-phosphorylated, that's A and B, right? And then we have the R and T states on top of those. Now, the B form does favor the T state. Okay? And the A form does favor the R state. But you can have both states for each of these forms. Now again, which one's phosphorylated again? Okay, so the A state is the phosphorylated versus it does favor the R state, okay, which is the more active. The B state is the non-phosphorylated, which does favor the T state, but can be in both. Okay. Does that make sense now? Fantastic. Fantastic. And that's the phosphorylations right there, in case you're wondering. Very good. So the role of the glycogen degradation in the liver is actually going to be to maintain the adequate blood glucose levels. It's going to result in the A form being the default state in the liver. The liver is going to phosphorylase A form is going to exhibit the most responsive R to T transition. The binding of the glucose to the active site is going to shift the A form to the active R state to the less active T state. And that's what we're seeing right here, actually. This is the phosphorylase A. Remember, that's the more active form due to phosphorylation. Okay? And this is going to be your default in your liver because your liver is constantly having to give off glycogen glycogen, excuse me, to the rest of your body, right? And so this is your T state right here, and this is going to be binding your glucose. When you're not binding your glucose, you're typically going to be in your R state, right there. So the default form of the muscle phosphorylase is actually the B form, which is the less active form, because your muscle phosphorylase doesn't have to do that as often, right? It's not typically supplying your glucose to your brain, etc. So the phosphorylase is going to be active during muscle contraction. The AMP is going to activate the muscle phosphorylase B by binding to a nucleotide binding site, and it'll stabilize your R state, which will be your more active binding state. Your ATP and glucose 6-phosphate allosterically inhibit muscle phosphorylase and stabilizes the T state. And so your ATP is going to compete with your AMP on those sites. Your glucose 6-phosphate binds the ATP binding site and stabilizes the phosphorylase B. And this is what we're seeing right here. So this is the less active, non-phosphorylated form, okay? And this is the muscle one. And we need more granularity in the muscle, right? The liver doesn't actually use the glucose typically, okay? So if you're a bit low on glucose in the liver, not a huge issue, right? On the other hand, your muscles are going to use glucose fairly often, aren't they? Because you're going to be doing a lot of contraction and a lot of motion, okay? Yeah, they don't have to depend it only on like the brain does, but still. And so we have a more modulated form of this. This is actually your nucleotide binding sites right here that we were talking about before. And so the ATP or the two glucose 6-phosphates are actually going to work to convert that. And this is the AMP right there. So skeletal muscles actually can consist of 3 fiber types. You guys have learned about this, right? Yeah? No? Okay. Every now and then I wonder if I'm like the first one to break these things. Hopefully not. Type 1 is going to be the slow twitch fibers. These are going to be fibers that use cellular respiration. They're going to be powered by fatty acid degradation. They're going to generate ATP. Type 2, which is 2B, excuse me, which is your fast twitch fibers. These are going to be the ones that use glycogen as their main fuel. Type 2A fibers are going to be fibers that possess properties that intermediate between the other 2 fibers, and they'll be sort of like a mixture of the other 2, okay, at least from the biochemistry perspective. All right, so these are the biochemical characteristics of your muscle fiber types. Type 1 is going to have very high fatigue resistance. Mitochondrial density is going to be very high. Metabolic type is going to be oxidative. Myoglobin content will be high because we're pulling a lot of oxygen. The glycogen content is going to be very low, okay? Because we don't need that as much to power it. And remember, if you have glycogen there, what does that mean? Is there any consequences to that, or is just that like free? Okay. Do you guys carry a backup battery for your phone? No? Why don't you carry a backup battery in your pocket for your phone? Inconvenient? Why? Has size, right? Pulls your pants down. It's annoying, right? Yeah. And same thing here. Why don't you just want to put glycogen everywhere? Some space. Yep. It has a cost, right? It has weight, has space, has a cost. Yeah. And so, yeah, we don't need the glycogen as much. We're going to rely more on the metabolic pathways for aerobic, high oxygen content. We're setting ourselves up for that, okay? And again, type 1, these are the slow twitch fibers. We're typically going to be doing endurance stuff with these, right? So we have the time to flow the oxygen to them, okay? And so the triacylglycerol content is going to be very high. The glycogen phosphorylase activity very low because we're not depending on glycogen, right? The phosphofructokinase activity, also low. Citrate synthase activity, very high, because again, we're a citric acid cycle. The type 2A, this is going to be your intermediates, right? And so basically, it's going to be middle between all these things, okay? It's going to be a combination of the two in terms of activities. Type 2B, on the other hand, fast twitch. We're going to run out of oxygen, aren't we? Yeah. We're going to use up so much energy, oxygen flow can't keep up. So what do we need to do? Well, fatigue resistance low, because we're going to burn through our energy very quickly. Okay? Mitochondrial density low. Why? It's powerhouse cell, right? Yeah, we're not doing the normal cellular respiration where we would use the oxygen, right? Because we can't afford it. Okay? Kind of the same reason as why do you not put a nuclear generator in your car? You're not going to get a chance to use it, are you? No. The metabolic type, glycolytic. We're just going to straight up use the glucose that we store in the glycogen, aren't we? That's the majority of our energy that we're going to end up using. Myoglobin content, low. We're not depending on oxygen coming in. Myoglobin would have helped us get more oxygen in, aren't we? The glycogen content, high. Again, it's storage, but we just have to utilize that cost, right? Same reason you have to have a gas tank on your car, right? Because you can't account for having a wire hooking you up to the power plant. Yeah. Triacylglycerol content, low. Glycogen phosphorylase activity, high, because we're using glycogen. Phosphofructokinase activity, high. So the glucagon, this is going to be a peptide hormone that's going to be released in response to low blood glucose levels. This is actually the glucagon order right here. Epinephrine, typically called adrenaline, is going to be the hormone that's released during strong emotions. These major emotions tend to be things like fear, excitement, though low amounts they actually get released due to stress, and it's actually quite a ubiquitous response, apparently. Anyways, adrenaline, epinephrine. You guys know that fairly well, right? Okay, so the phosphorylase activity, a kinase is going to be the phosphorylase. It's going to be the kinase that acts on that enzyme. And this is going to be a regulatory enzyme that catalyzes the phosphorylation of a single serine residue in each subunit of your phosphorylase. This will yield the phosphorylase A, which remember is the prominent form in the liver. It's a more active form, right? This is catalyzed in response to the glucagon or epinephrine. Both the liver and the muscle phosphorylase can be covalently modified. Phosphorylation is going to move a peptide loop of the active site of the B form. It's going to render the enzyme more active. By the way, has anybody ever wondered why okay, so normally if you have a shot of epinephrine, what's going to happen to you? You're going to become very active, right? Very high energy. But has anybody ever seen somebody that has a extended period of time where they've had a lot of adrenaline rush? No? Anybody ever been under stress? I know you have, because you've been in my class. Ooh, look at you all. You're looking at me like, oh, Dr. Clausen, I hate you sometimes. You don't joke about that, man. Under stress responses, you have low levels of adrenaline coming off all the time. And so notice what happens after a while. Stress at first makes you very high energy, right? Stress after hours of it, makes you tired. And that's because part of what's happening is you're switching off onto a glucose pathway. You're running through your glycogen stores. You're running through that glucose, right? And those pathways are not exactly the highest output of energy in the long term, are they? They're kind of inefficient. And so you're doing a more inefficient pathway. Okay? And you're running through your energy stores. That should have been that immediate boost. Okay. The phosphorylase kinase, this has a subunit composition. The subunit composition is alpha, beta, gamma, delta, 4. 4 for the whole thing. The alpha beta subunits are going to be phosphorylation targets. Your gamma subunit contains the active site. Your delta subunit is the calcium binding protein, calmodulin. Notice again, those motifs are showing up over and over, right? The calcium bonding modulin, that's literally a delta subunit. You actually have to kind of wonder, honestly, some of these, I kind of wonder why they don't just standardize the subunit format. Like calmodulin always shows up as like capital CAL or something. Yeah. Anyways, maybe someday, right? Maybe you guys can do that. There you go. You can standardize it for Dr. Clausen, so people don't have to remember this one's delta, this one's beta, they're calmodulin. It's always cal. So the activation of your phosphorylase kinase is going to be initiated when the calcium 2+ binds to the delta subunit, calmodulin. The maximal activation is going to occur with the phosphorylation of the beta and the alpha subunits by the protein kinase A. Again, those are your phosphorylation targets. So this is the phosphorylase kinase right here. This is the inactive form. Again, there's the alpha, there's the delta, gamma, the blue things in the back are the betas. Okay? Notice the alpha and betas look very similar, right? Again, the phosphorylation targets. Okay, so calcium's going to come along. What's it going to bind? Delta, calmodulin, right? And that's right there. You can see the CAs in it. So that's something like a nerve impulse, muscle contraction. It could be a hormones. Remember some of the hormones, 7-TMs, release calcium. It could be any of those things, right? So we get calcium bound to this. This is going to be the partially active form of this kinase. So what's going to happen next is pKa is then going to fully activate this by phosphorylating the alpha and beta subunits. And this is typically hormones that do this and put this into full activation. Okay? And that's that same pKa hormone pathway we talked about before. Notice that some of those pathways we talked about would have actually done both, because the calcium was part of the pKa activation. Okay? So, I know we're wrapping back around finally, aren't we? You guys are so happy you studied that, right? Okay, so fully active form. In this case right here, we now have the fully active form. This is the form that's actually going to do this phosphorylation on our phosphorylase alpha betas. Okay, so we're going to phosphorylate our phosphorylase beta to phosphorylase alpha right here. And I know that it gets kind of annoying sometimes because we phosphorylate a phosphorylated, phosphorylated, phosphorylated. And it just keeps going, doesn't it? Yeah. But that's a very typical motif to activate and activate different enzymes, because we have phosphorylates that can move off and on ATPs. We have the phosphorylate potential energy, right? So the protein kinase A, the pKa, this is going to activate the phosphorylase kinase. It's going to turn and activate the glycogen phosphorylase. So right here we're seeing the glycogen breakdown controlled by the combined effects of the multiple hormones in our tissue-specific manner. Fasting is going to be a low glucose conditions. Typically fasting is actually denoted by that low glucose condition. That's what they typically call that and require that. It's kind of interesting that fasting cannot be just 6 hours of non-eating. Okay? And then your glucose levels drop off precipitously from there and then sort of reach a minimum. So your liver here is going to have your glycogen going to glucose. Note, by the way, your lactate from the, oh, we're done. Okay, let's just stop on this figure and we'll start from here next time. Yeah, because this is a kind of loaded up figure. And we'll have the pathway after this, and I want those to be separated. All right, you all have a wonderful day. I will see you Monday. Actually, have a wonderful weekend.